Okay, so I think we're on. Hi, everyone. I'm going to do my presentation. I just need some hello or hi from anyone. Just make sure that it's now working. Okay, great. Thank you. Okay, I'm going to focus on my screen, and Scott, please let me know or text me my cell phone if there was something wrong with the presentation. Okay. My name is Milad Lankarani, and I'm a scientist at the Cranfield Research Institute, University Health Network, and assistant professor at the University of Toronto Department of Biomedical Engineering, and an affiliate scientist at Toronto Rehabilitation Institute. As I'm thinking, I might go a little bit over time. I try not, but I tried. I try not, but I'm going to thank my lab members. Our lab, neural system and brain signal processing, is based at Cranfield Research Institute, University Health Network. I would like to thank my mentors, Professor Francis Skinner, Professor Miloš Popovic at TRI, University Health Network, and Professor Steve Prescott at SickKids. I would like also to thank Cranfield Research Institute, University Health Network, and some other granting agencies like MyTax, NSERC, and CMC for supporting my research. I should mention that the result of this presentation, some other works related to the whole modeling of deep brain stimulation, cellular mechanism, network mechanism, is a result of collaboration with my great and fantastic collaborators, Dr. William Hodgson at the Cranfield Cranfield Research Institute and University Health Network, and Dr. Luka Milosevic, again, Cranfield and University of Tübingen. Okay, I start with the motivation of this presentation, which is on understanding how DBS modulates information processing, and mostly in the network level. But I'm going to start from the effect of the DBS on the single neurons, and to state the problem that we know that DBS alters neuronal response, and experimental evidence showed that DBS actually modulates synaptic plasticity, the way that neurons communicate with each other. And if we just imagine that we have a simple neuronal circuit, it can be based on ganglion network or any other substructure in the brain. When we have the deep brain stimulation, we not only modulate or change the neuronal activity of single neurons, but also the way that neurons are communicating. So everything in the network might be affected because of the deep brain stimulation. And in this presentation, I'm going to discuss on that a little bit. For sure, it's not fully covered. But the main objective is to understand or to know or to estimate the neural response of single neuron, and also the response of or the changes of the synaptic plasticity affected by deep brain stimulation. Okay, as I mentioned, the objective is to understand neuronal response and synaptic plasticity, or functioning underlying synaptic plasticity. Here I just showed the electrode, DBS electrode, that my collaborators used to do this simulation and record from human cells. And you can see that we have simultaneous stimulation, which are pulses, and usually for clinical application, we use high frequency, 130 hertz. It depends on the area of the simulation, and this is kind of recording, as you can see in this. Just to clarify, this is single unit recordings showing the spikes. Okay, and experimental evidence, as I mentioned, demonstrated deep brain stimulation modulates synaptic plasticity. We have two types of plasticity, short-term and long-term. I also covered the effect of DBS on short-term synaptic plasticity. On the right, I just try to make the problem as a black box where we have the simulation electrode here, and I assume that, for instance, this is a very small area in the brain, let's say STN or thalamus or wherever we have a deep brain stimulation, and we also have recordings, we access to spikes, and I simplified this black box to two building blocks, synaptic function and neuronal response that I'll show you in the next slide or slides why I do this. But two main unknowns are what are the neuron response and synaptic function due to changes of this stimulus, which is deep brain stimulation pulses. And what I mean by changes in the stimulus, I mean the frequency of the deep brain stimulation. Okay, in this presentation, I'm going to give you a brief background on short-term synaptic plasticity and also linear time invariant systems that I used here. It's a very simple model, and I should justify why I'm using this. This is hard work for this presentation, but it works okay. And then given 1 and 2, I'm going to identify what I call impulse response function, underlying neuronal response, as well as the frequency response of synaptic function when we have deep brain stimulation. Okay, for just quickly go for short-term synaptic plasticity, because I want to spend most of my time for the modeling of the or estimating the frequency response of the plasticity. Here we assume that we have deep brain electrode, and this is called where we assume that we are targeting this neuron, and so this is called stimulated neuron or stimulated nucleus. And I'm not going through different hypotheses how it works, but as Professor McIntyre mentioned, that where we have DBS, we have some spikes because of these DBS pulses induced on the axon of this stimulated neuron. This can go either orthodermically or antidromically, and wherever we have spikes in general, either because of DBS or because of normal activity of the neuron, we have, let's say, so we have spikes here. Here, because of the synapse, if the neurons are connected, we have some changes in the postsynaptic potential, and you can see that for each single spike, we have some postsynaptic potential. Now, by synaptic plasticity, we mean that the amplitude of this postsynaptic potential is changing due to the rate of these presynaptic spikes. Okay, so it might increase where we call it facilitatory synapse, or it might decrease that we call it depression. So we have different types of synapse, but if the amplitude is changing depending on the rate of the presynaptic spikes, then we call it synaptic plasticity. Okay, and we have this very well-known model for synaptic plasticity. It's called Sodex's-Markov model, including 3 differential equations. The last one, which is I, represents this, what you can see as the postsynaptic current or postsynaptic potential in general. And these 2 hidden variables represent the, let's say, utilization of the calcium and neurotransmitter release, and this is the depletion of neurotransmitter. I'm not going through the detail of the biophysics of that, but what we can do with this model, we can replicate the plasticity in different type of synapses. Okay, the second background is on linear time invariant systems. In linear time invariant systems, any system that satisfies 2 conditions of linearity and time invariance is called LTI system. And for LTI system, what we should know, or what we want to know, is the impulse response. Okay, and we can use simple convolution techniques to represent the inputs and output relationship. And so the simple concept of this LTI system is that given any inputs, if we have the impulse response function, we can generate and create the output. And frequency response is just the Fourier transform of the impulse response. Okay, I think this is the first point that you might ask, okay, why LTI system? Do we use the LTI system to represent neuron response? As we know, the neurons are not linear. Okay, so this is not probably a good approximation. But I just wanted to mention that we do not necessarily want to replicate the spikes. We want to estimate the rates of the spikes. And this is just a simple simulation showing that if we have the input and if we plot the rate of the spikes, as you can see here, the relationship between this input and the rate of the spikes can be a very simple linear time invariant system. Okay, so we can rely on the LTI system. It's not the neuronal response in the sense of the spikes, but it can be the rate of spikes. And the reason I used the block for separated neuronal response and synaptic function for DBS was just simple. Again, we have different hypotheses how it works, but simply two main hypotheses is that the spikes are generated because of the DBS pulse on the axon of the simulated neuron and target postsynaptic neurons, or we have some activation of presynaptic terminals. Again, these are the axons that target the stimulated neuron. Okay. Anyway, for any hypothesis, we can consider that there is one functionality here between, there's a synaptic function, and the other one is the neuronal response to the input of DBS pulse. So for each single DBS pulse, we have some changes on the synapse. You can also consider this synapse, doesn't matter. And also we have some changes of activity in the neurons, okay? It's because, anyway, we either record from here, which is the result of this synaptic function and the neuron response of this neuron, or we stimulate here and record from the postsynaptic target of this neuron where we have plasticity, synaptic function here, and the neuronal response of this target neuron. Anyways, we can use this, and now you can imagine why I use LTI system, because in LTI system, doesn't matter. You can switch these two block diagrams. Okay, so this is just mentioning one approach which was done, Dr. McIntyre Group 2019, that they consider, so if we have deep brain stimulation in the thalamus and we record from the M1 and the cortex, this is how this looks like, and each here, each row represent the neuronal response or the spikes of each single pulse. And it was high frequency deep brain stimulation, I think 130 hertz. This is why in the X domain you see around 7 point something millisecond, that together with this 130 hertz pulse, this would be, the total would be 1 second. And this is kind of raster plot. What was shown here was that with this approach, which is a very well-known approach in computational neuroscience, we have a model, and we try to use a model of, for instance, plasticity or neuron models, and we try to construct the neuron or network and see how good we can replicate the experimental data. And what they showed is that by including the model of plasticity, we have this representation in computational models, which is much better than these, where we have deep brain stimulation. You can see the average frame rates, if you compare B and C, B is kind of reduced and is, closer to experimental data. So we use this approach in collaboration with Luca and William, and also my master student Alireza, and we come up with some interesting results, both for understanding a mechanism of how DBS works in cellular level, plus estimating the parameter of synaptic plasticity. But in this talk, I'm going to use another approach to this question. So it's not very straightforward kind of building the neuron models in computational neuroscience. Just quickly go to this slide, which is the way that we get the experimental data. It's from mostly the DBS are located in the thalamus. So we get the recordings from ventral intermediate nucleus, WIM, or reticular thalamic neurons. And so this is how they look like. We have different DBS frequencies. Okay, so here at the bottom you see different DBS frequency, and these are the pre-stimulus histogram or PSH of the recordings. Okay, so we have different frequency of DBS, and we have the neuronal recordings. Okay, now our approach that I'm going to present here is inspired by neural coding, and very similar to very basic concept of the spike trigger average, where we have an image or any stimulus, and we have recordings, and we look into the most probable feature of the stimulus preceding the spikes, and we do the average. And according to this kind of average, or what is called spike trigger average, we can go back and reconstruct the stimulus. Okay, so this is called neural decoding, very simple way of neural decoding. But I use this strategy in different way to go in a forward way and to do neural encoding. And the strategy is that we have pulses, okay, and I'm trying to emphasize on low frequency DBS pulses because the effect of plasticity is not significant. I'll back to this, but we have pulses and we have recordings. Okay, again, if I plot the response to each pulse, okay, the spikes or neural recordings of each single pulse as a raster plot, and if I do the average, what I'm going to do is very similar to the spike trigger average, but instead of going back to the stimulus, I'm going to the response, and I'm looking into the most probable the most probable function of the neurons responded to these repetitive pulses. Okay, and what happened? We get something like this that I'm calling this as the impulse response function of the neuron. Okay, you might ask, okay, what about the synapse? Synapse is there? Yes, of course. I plotted the frequency response of synapse. This is the main thing I'm going to show you how I calculate this in this presentation, but just trust me that in low frequency, the effect of this synaptic function, the effect of plasticity is negligible, and so we can trust and this is the neuronal response of the neuron. Okay, and the best way you can ask me, okay, how do you know that this is correct? I'm going to predict what happened, and this is just showing the way that we get the impulse response function of the neuron given low frequency DBS pulses, and this is for VIM, and this is for RT neurons, and you can see this is the specific shape, and this is the baseline. Okay, now assuming this is the impulse response function of the neuron, what you should do? Very similar to the neural decoding, I had spikes. I go back and reconstruct the stimulus. Okay, here I have DBS pulse. I calculate the impulse response function of the neuron. Okay, assuming that the function underlying plasticity is negligible in low frequency DBS, and I'm going to predict neural recordings. Okay, so let's do it in low frequency DBS 5 Hertz. Black is actually the PSTH of the RT neurons. The result for RT and VIM are very similar, and the black is the experimental data. Okay, this PSTH of the recordings, and the blue is the estimated one. The way that I just convolve DBS pulse with the impulse response function that I showed you here. Okay, now if I go to 10 Hertz, I again get very good kind of match between the estimated or predicted response and neural recordings. Now, when I go to 20 Hertz and 100 Hertz, this happened. The response of the neuron is decaying, as you can see here. If I go to higher frequency, it's going to baseline, and if I continue, you will see it goes and stay here in the baseline. But the prediction, which is just simple convolution of DBS pulse and the impulse response function, goes steady state value and stay here. Okay, we see this mismatch. Okay, so if you compare this with this previous figure, you see that, okay, there is something wrong. There should be something. That something, we call it as the synaptic function. Okay, we actually consider that there should be something between what we predict and what we observe that indicate the functionality underlying plasticity. Now, what should we do? How we calculate this? We should notice that this synaptic function or plasticity is dependent to the frequency. Okay, to the frequency of the presynaptic sparks. In this case, you can consider this as deep brain stimulation pulses. Now, what I'm going to do, I'm going to calculate the frequency response of this synaptic function. And how do I do that? Simply, if you have a system that you can stimulate, if you use, let's say, 1 hertz stimulus and you get the output, okay, we just simply go and calculate the power of the response and divide this by the power of the input. Okay, if I continue to do this and for different frequency, I can make the frequency response or the power of the Fourier transform of the HT. Okay, now I have done the same thing for human neurons. Okay, what we have is that for 5 hertz, for 10 hertz, what we can do is that we are going to calculate the power of the response, which is in red. Okay, and power of the response, which is in blue, and the power of prediction, okay, which is red. And you can see that when we go to higher frequency, we see a big difference between or mismatch between the power of the recorded spikes, which is blue, and the power of the prediction, okay, which is simple convolution. Now, what we can create is the frequency response or the power of the Fourier transform of the synaptic function, or whatever we need there to compensate this mismatch. And interestingly, we see some kind of low pass filtering function of this frequency response, indicating that when we go to high frequency DBS, okay, the power of this synaptic function decreases, which exactly equivalent to depression. Okay, now what I'm going to do, I'm going to use the Sodexmark model, and for different type of synapses. I just got the values of these synapses from the literature in animal for glutamatergic synapses. And we have 3 different types, depression, facilitatory, and pseudolinear. In the red, you can see just pulses, okay? And black, you can see the synaptic current or a synaptic potential, okay, for different type of synapses. Now, when I have, let's say, 4 hertz, you can see, forget about transit because what we care is just the stationary part. What you can see is that there won't be too much changes between the postsynaptic current and the pulses. Okay, everything is here is normalized. But when we go to higher frequency, we see that for depression, or we have different behavior for depression, facilitation, and pseudolinear synapses. But interestingly, when we go to higher frequency, all of them are decreasing. Okay, now using the same strategy, we can rebuild the frequency response of these 3 type of synapses. Okay, so these are the model. Okay, I use the stimulus, which is pulse, different frequency, and I calculate the power of the response in that specific frequency, and that's it. Okay, so we see that for depression, facilitation, and pseudolinear, we have this kind of shape. All of them are kind of low pass, but you can see that for instance, for facilitation, we have a wide dynamic range in frequency. But when we go to higher frequency, all of them going down. So this is my last slide. Going back to before going back to the question, is that what we could get without any, let's say, model or pre-assumption about the model was that we could recreate, or we could not recreate, we could extract the frequency response of the synaptic function, which is related to the synaptic plasticity induced by deep brain stimulation. Okay, and when we use the model, simple model of Sodexmark model, we found that these models also show some similarity between these, but for sure in the neurons we have different type of synapses. So combination of these will create this frequency response of the synapse that we could calculate. I'm going to capitalize on the important role of these synaptic plasticity and how DBS is modulating that, because on the x-axis, actually, we have DBS pulse. So this frequency is the frequency of deep brain stimulation. And if we change that, we are going to different situation for transmitting information. And if we assume that deep brain stimulation is stimulating not simultaneously, but each individual substructure, let's say SDN, SNR, GPI, or thalamic neurons, we actually playing with a dynamic range in the frequency domain of information transmission in the whole network, which is super important in building the neuronal circuit underlying, let's say, for instance, basal ganglia network underlying the effect of deep brain stimulation. And yeah, that's it. I'm going back to and I stop sharing, and I'm happy to answer your questions. Okay. Okay, we have one question. Just for some clarification, is the impulse response function a time varying average of the spikes over a time window? The answer is yes, but since we go to and we use a lot of these pulses and we get the average of many kind of trials, then we can consider this. It is time varying in the sense that the behavior of the response changing over time. But in terms of the system consideration, I can call this as linear time invariant system, because given the input and using that impulse response function, which is for sure varies over time, but with that impulse response function, you can predict the output to some extent, for sure lower frequencies. Okay, so okay, so we don't have any more questions, and I'm assuming that this is not a bad sign. Okay, great. So, okay, so we have another question. Okay, start. Does the time invariant nonlinear black box identification approach make sense, for example, to account for synaptic plasticity at higher frequencies? Yeah, exactly. So thanks for this question. So this presentation, I just wanted to make the ground for using very simple linear time invariant system to show how we can extract. So the whole frequency response of the synaptic plasticity that I showed here is just a proof of concept. This is what people already use synaptic depression model, and they could recreate, as I showed in a slide, the group work in a group led by Dr. McIntyre. So this was a very simple proof of concept, but in a different way. And for sure, we can use a nonlinear system. It is totally fine. We can use even we can introduce some randomness in the model to better represent the spikes or the rate of spikes. And yeah, for sure. So the answer is yes, definitely. Okay. How did we can? Okay, so let me go back. How can you pick apart the effect of different neuron synapses in the network in your frequency response function? Well, yes, true. This is a fair point. I really can't. The only thing is that based on the simulation and recording, we know that the recording is from that specific single neurons spikes. And then we have some hypothesis that so it's based on hypothesis that the effect of that neuronal response is coming from the neuron and the way that deep brain stimulation affected on the presynaptic terminal of that neuron. Okay, so this is the way, but systematically, it's really hard to answer this question if I understood it well. Okay, thank you so much. But I'm happy to continue to talk probably in panel discussion. Thank you, everyone. I'm going to end broadcasting of this talk, and we are going to have Tawfiq in the next session. Thank you.